but we're going to go ahead and get started. Very pleased today to have two folks with us from Hampton Roads Sanitation District, aka HRSD, and down in their neck of the woods, all you have to say is HRSD and everybody knows what you mean. But in other parts of the state, I try to remember to say Hampton Roads Sanitation District first, because not everybody knows what HRSD is, even though it's been around for 70 years.
85 years. Our anniversary was just about a week and a half ago.
Okay. All right. Wow. Yeah, I grew up in Hampton, so
Oh, wow.
it predates me, I'm happy to say. But okay. We have with us today Jamie Heisig-Mitchell and Chris Burbage. Chris's camera's not working, so he will just be speaking to us as a voice from out of the dark. And Jamie, if you want to take it away. Well, no, before we do that, let's go around and introduce everybody.
Okay.
All right, I'm going to call on folks based on how they're sitting in my screen. Sarah.
Sarah Parmalee of the Piedmont Environmental Council, covering Culpeper and Rappahannock.
Ken.
Ken Ohm, John Marshall, Soil and Water.
Deirdre.
Deirdre Clark, John Marshall, Soil and Water, covering Fauquier County.
John.
John McCarthy, Senior Advisor, PEC, covering whatever they let me play with.
All right, Larry, that's you. You looks like you're mobile.
I am Larry Zaragoza. I live in Northern Virginia.
Okay, Michelle. Thanks, Larry.
Hello, Michelle Edwards with the Rappahannock-Rapidan Regional Commission.
Stuart.
I'm Stuart Overby with Don't Spread on Me in Albemarle County.
All right, Dan.
Morning, everybody. Dan Schaefer with Allegheny Blue Ridge Alliance. We cover Western Virginia and Eastern West Virginia.
Okay, Barbara. She's got to get off mute on an iPhone.
Good morning. Good morning. I'm here. Barbara Walsh. I'm with the Environmental Protection Network, located in Rockbridge County.
Okay. We are far afield today. Ian.
Hey, good morning. With Dominion Energy, first time joining the call.
Glad to have you here, Ian.
Thank you.
Brian.
Good morning, Brian Bays, Virginia Department of Forestry, Regional Forester covering our Eastern region.
Thanks, Brian. Cassie. Right.
Hello, Cassie Smith, Troutman Pepper Lock and VBC.
Thank you. Brianna.
Hello, Brianna Heath, George Washington Regional Commission.
Another interloper, Robert.
Morning, Eldon. Robert Crockett with Advanced Strategies for a variety of clients, including VBC.
For a variety of clients, I like that. I understand that. You know, it's kind of like the what was it, Dr. Zeus with all the hats. Yeah. And we have a phone number, 585 area code.
Yep. This is Evan Isaacson with Chesapeake Legal Alliance. Thanks for having me.
Great. Thank you. And just coming in is Pat Calvert with American Rivers. Pat, you want to introduce yourself? You just caught in at the very end of the introductions.
Good morning, Pat Calvert, American Rivers.
Good to have you. Okay, with that, Jamie, I'm going to turn to you and you can share your screen.
All right. Now we practiced this before, so let's see if it'll still work. All right, let me go to slideshow.
We're getting there.
There. Click that again.
All right.
Okay, so are you seeing it in full screen mode?
Not yet.
Not yet.
There you go.
Is it all set?
Yep.
All right. Well, good morning and thank you for your time. Really appreciate the opportunity for my colleague, Dr. Chris Burbage, and I to share information that is Virginia specific. There's a lot of important research projects going on related to PFAS and biosolids. And fortunately, we do have some Virginia specific connections and information to share. So Chris and I are officially representing HRSD. HRSD is Hampton Roads Sanitation District, but our service area actually extends well beyond Hampton Roads. So we're actually trying to lean a little bit more into just the HRSD part so that we can kind of start to express that we're much more than just Hampton Roads now. We also participate in the Virginia Biosolids Council. Chris and I are also members of the VBC's research committee. We're also members of the Virginia Association of Municipal Wastewater Agencies. So that's VAMWA. We're on their biosolids committee. I'm past chair of the National Associations of Clean Water Association, National Association of Clean Water Agencies. I'm a past chair of their biosolids committee as well. So from a wastewater perspective, we've been engaged in a lot of research, a lot of discussions. And we're here this morning to talk to you very specifically about the research that we've been participating in, and just have to acknowledge that we're not alone in this at all, that there are other Virginia utilities that have been participating in this work, and the Virginia Biosolids Council and other utilities have also funded a lot of this work as well. So before I talk research, though, I do want to orient you to us. Let me go ahead and I need to minimize you all so I can see my screen a little bit better. So HRSD is a regional wastewater utility. We serve 20 cities and counties in Southeast Virginia. As I mentioned, we go well beyond Hampton Roads at this point. You can see we have facilities in the Eastern Shore, all the way up to King William County, and we serve out west to Surrey County on the south side as well. Across that service area, we serve about 20% of the population of Virginia. We operate 14 treatment facilities currently, and they have a combined capacity of 225 million gallons per day. We're actually a political subdivision of the Commonwealth. That's how we were formed 85 years ago. We were formed as a regional entity, really focused more on those metro areas of Hampton Roads. But with that working as a political subdivision, being established as a regional entity, we're also overseen by 8 governor-appointed commissioners. And ultimately, all of that allows us to really think more regionally and not just focused on individual cities. And that regional thinking allows us to really think about some of the more complex water and wastewater challenges that affect a large area. And in terms of implementing innovative solutions, we have a really active research and development department, and we participate in a number of research projects throughout the country. We're engaged in 32 active water research foundation projects right now that extend well past PFAS. And I know that pretty much everyone on this call has heard about PFAS for quite a while, the per and polyfluoro aqua substances. But this is just the obligatory slide to remind us that its ubiquity has really integrated with all aspects of our lives. So it's everywhere, unfortunately. So land application is important. Let's see. So land application does remain a viable management option, but there are three options that are really important for the daily production of material. 3 primary management options are land application and recycling, incineration, and landfill. I'm going to touch on each of those. So of the 3 options, land application and recycling really allows us to return that renewable source of nutrients and organic matter back to the soils, provides an opportunity to provide improved soil health, provides carbon storage benefits, provides a slow release nitrogen fertilizer, and really reduces the reliance on commercial fertilizer. Incineration is another management option. In the state of Virginia, there aren't a lot of facilities that have incineration capability. There's less than a handful of wastewater utilities. I think there are only 3. HRSD is one of those utilities that has incineration capability. And then landfill is another management option for biosolids. So landfill capacity is also limited. There was a recent survey of landfill capacity commissioned by Alex Renew, because all wastewater agencies are trying to figure out, how to best manage this material. So Alex Renew commissioned this landfill survey and identified that Virginia landfills just really lack the capacity to manage all of the biosolids generated in the state at the current acceptance rate. So that doesn't even count material that might be coming in from outside of the state. So with 14 facilities that I just pointed out, 14 facilities scattered across Southeast Virginia, we've used all three approaches. In many ways, that mirrors what's going on across the state. The state uses all three approaches, and reliance on a single approach is particularly problematic. And so for us,
Jamie,
yeah,
quick question for you before you go
Yeah.
forward. When you talked about incineration, you said HRC.
HRSD, we have one. We have a couple of incinerators. We do have the capacity to incinerate.
Okay, so it's your incinerator.
Yeah, we do. Yep, we have an incinerator. We have a couple of incinerators. And then there are a couple of other utilities that have incinerators as well.
Okay, thank you.
Yep, you're welcome. So as I mentioned, we do all of this. The state does all of this. So we kind of, because of our size, we kind of mirror what's going on in the state. So we really try to prioritize options that support a circular water economy so that we can take a recovered product from our wastewater treatment and really put it to productive use. And in that case, we're thinking about land application and composting, and that's a strategy that HRSD employs. So land application is still considered safe and a safe and viable management strategy. And I think that's important to consider, particularly since Virginia's required management practices for land application exceed federal requirements. And I think that's an additional protective measure that Virginia provides. I think they have strong regulatory oversight of the program. And I think in addition to some of the other restrictions that they have, I think the ones that are key for this is that we have additional setbacks beyond what the federal program requires that are in place to protect waterways and private wells. I also want to note that based on the data that I've seen, concentrations in municipal biosolids that don't have industrial sources of PFAS really largely reflect the daily exposure from our households. And I think this makes sense because, most facilities are treating largely domestic waste. And so our residential exposures are getting reflected in the biosolids. And so when we look at successful programs around the country, what we've seen is that source control efforts that really focus on reducing the industrial inputs of PFAS have been pretty successful at bringing down PFAS concentrations. And I do want to also note, I think this third bullet is important as well. HRSD and a lot of other Virginia utilities support a structured PFAS monitoring program of that biosolid source material so that DEQ can get useful information about the PFAS characteristics of the biosolids. This is what we've seen, again, for those successful programs around the country. And you've heard people talk about Michigan, probably, if you've been following the biosolids discussions. Really focusing on understanding and characterizing your biosolids initially is important. And then focusing on source control where you have those outliers. When we do the monitoring of the biosolids, I do want to also point out that it's really important to do that monitoring at the source facility, so at the generating wastewater facility, to get an idea of the concentration of PFAS in the biosolids. Once it gets out onto the field, there are just too many current and historical PFAS sources that could be making that characterization inaccurate and difficult to really tell where the PFAS is coming from. So if you really want to understand what the PFAS characteristics are of the biosolids, then you really need to do the monitoring at the generating facility. And this slide is just a super brief introduction to some of the research that's going on in PFAS. So wastewater utilities are continuing to invest in research where HRSD is participating in some of these. Other utilities around the state and around the country are participating in some of these. This is a short list from the Water Research Foundation of projects that I thought you'd be interested in. We're not going to go into these because they're ongoing projects, but they're ones to watch out for. The first one is this PFAS One Water approach to managing PFAS pollution. That's scheduled to complete in 2027. And that's when we talk about a one water approach, we're talking about really being holistic and thinking about wastewater, stormwater, drinking water sources, and really trying to manage that PFAS pollution. The other one here is understanding thermal processes to mitigate PFAS. So we're looking at thermal destruction techniques for PFAS mitigation. Another one that HRSD is participating in, should be wrapping up soon, is looking at PFAS through sewage sludge incinerators, so kind of linking that incineration piece. And then the last two, again, just other items, other areas of research to really try to mitigate and control PFAS that ultimately end up in the biosolids. So these are just a few projects. There are about 20 projects going on right now at WRF that are related to PFAS alone. So right now I'm going to go ahead and turn it over to Chris, and Chris is going to talk specifically about 4 projects that have Virginia connections, and we'll be able to provide some Virginia data that I think you'll be interested in to really kind of hone in on what's going on in the state. So I'll go ahead and start on Chris's slides.
Okay, thanks, Jamie. Over the past 5+ years, there have been several major PFAS studies conducted here in Virginia. These studies have been conducted as part of local, regional, and national efforts that were led by academic institutions, water sector NGOs, the Virginia Biosolids Council, and individual utilities like HRSD. And as Jamie said, these are 4 projects that HRSD has taken part in here, specifically in Virginia. Next slide. Of the 4 projects, 3 of these projects have taken place specifically at our HRSD Atlantic Treatment Plant. And we have an accompanying agricultural research farm. It's the Progress Farm, which is adjacent to the plant. And this facility is out in Virginia Beach, sort of southeast Virginia Beach, right near the ocean front. The treatment plant itself is a 54 MGD plant. It produces 20 dry tons per day of biosolids. And up until 2020, the facility produced exclusively Class B biosolids. And then in 2020, we initiated the thermal hydrolysis process. And since 2021, we've been producing Class A biosolids at this treatment plant. The Progress Farm itself, you can see the aerial photograph on the right, is a 180-acre agricultural tract. It's unique in that it has a groundwater monitoring system. There's 6 wells that are scattered around the perimeter of the field. It also has stormwater collection sites. And most importantly, it has a historical data set that goes along with it. HRSD has been doing research on this agricultural plot for actually over 40 years now. So it presents a unique opportunity to do biosolid research. Next slide. The first study I'm going to talk about is Dr. Rob Hale's EPA-funded research project. His project is focusing on the identification and quantification of chemicals associated with microplastic particles. And these microplastic particles can be found in biosolids. His project is focusing specifically on how operational changes in biosolid processing impacts the fate and transport of microplastic chemical constituents. There's also a component of this project that looks at the bioavailability of these chemical constituents after biosolids are land applied. And HRSD is participating in this research by providing biosolid material and other in-kind service efforts. Next slide. This project, while not specifically related to PFAS, microplastics and their chemical constituents are still a concern in regards to biosolid land application and chemical fate and transport. Microplastics enter wastewater treatment plants through collection systems servicing both domestic and industrial sources. These microplastics can be both chemically and physically transformed as the particles move through the wastewater treatment plant process. And microplastics leave wastewater treatment plant facilities either by final effluent or biosolids, depending on their specific physical and chemical characteristics. And I would like to note that the above information that you see on the slide it comes from Dr. Hale's research proposal abstract. Due to significant analytical difficulties, this project has experienced a number of delays. These analytical methodological delays are known problems when attempting to quantify and identify microplastic particles and their chemical makeup. But in the meantime, HRSD is going to continue to support the Hale Lab with in-kind support for this project. Next slide. The next study that I'm going to talk about is it's the WRF 5082 project. This project covers wastewater, drinking water sourced from surface water, and drinking water sourced from groundwater. This project focused on the one water concept. For example, which industries are emitting PFAS, analytical methods. It covered a lot of components in PFAS risk assessment. A guidebook was published from the work performed in this study and provides a step-by-step sequence of events that inform the reader on finding and eliminating PFAS sources. This study was led by Kyle Thompson from Corolla Engineers, but involved numerous PFAS researchers from across the country, including here in Virginia. And HRSD was a full participant in this research project. Next slide. Going a little bit deeper into the WRF 5082 study, the study provides an essential context for policymakers to understand the inherent circularity in our nation's water systems and the resulting impact on PFAS cycling. While individual utilities can take actions to prevent PFAS from entering their specific systems, the ultimate solution at the national level lies in reducing PFAS use at their primary sources, which is consumer and industrial products. Next slide. Okay, the next study is a study that came from Dr. Ian Pepper at the University of Arizona West Center. Dr. Pepper is the director of the Water Energy Sustainability Technology Center at the University of Arizona and is a leading national collaborative PFAS project. Dr. Pepper's leading this project that I'm going to talk about. Dr. Pepper's research historically has focused on the fate and transport of pathogens in air, water, soils, and municipal wastes. Dr. Pepper is a professor in the Community, Environment, and Policy Department in the University of Arizona College of Public Health, as well as professor in the Department of Agriculture and Biosystems Engineering. He's also director of the National Science Foundation Water Quality Center at the University of Arizona and a fellow of the American Association for the Advancement of Science, the American Academy of Microbiology, the American Society of Agronomy, and the Soil Science Society of America. Dr. Pepper is also a member of the W5170 group, which is a multi-state research project associated with the U.S. Department of Agriculture that focuses on the beneficial reuse of biosolids. The group's research is heavily focused on the study of PFAS, and most recently, he was a member of the EPA Science Advisory Board Biosolids Panel, which was charged with evaluating the framework for the sewage sludge chemical risk assessment. This national collaborative PFAS project was funded by both public and private entities and was designed to evaluate the incidence and mobility of PFAS in soils. And I'd like to add that the Virginia Biosolids Council helped fund the Virginia portion of this work, with HRSD offering in-kind sampling and analytical services. DC Water provided their Bloom biosolids as a designated exceptional quality biosolid material, which we used for the study at the Virginia Tech AREC in Warsaw, Virginia. Next slide, please. So Dr. Pepper's study can be broken down into two phases. Phase one of this national study created the largest U.S. data set of soil PFAS concentrations resulting from land application of municipal biosolids. The study consisted of 23 field sites located in 17 states across the United States, including Virginia. This portion of the study measured PFAS concentrations across multiple matrices such as soil, biosolids, groundwater, and stormwater. These measurements were collected over 12 months post prescribed land application, and this study was completed earlier this year, and the results can be found. There's a web link at the bottom of this slide, and that'll take you to his publication or his report on phase 1 of the study. Phase 2 of the study is currently ongoing, and this phase of the study is investigating the potential for crop uptake of PFAS analytes. And we just finished the fall soybean harvest out at the Warsaw AREC, and so those samples have been submitted for PFAS analysis, and we don't have any data back yet on that. Next slide. Okay, so this slide just kind of gives you a little bit of an idea of the study design for this national collaborative study. This is an aerial view of the Warsaw site, but all the sites across the country followed the same study design. So the study plan consists of dividing a 1-acre plot into 3 sections with a control plot where no biosolids were applied, a low biosolid application plot, which was approximately 5 wet tons per acre or 0.5 times the approved agronomic rate for this specific field. And then a high biosolid application plot, which was approximately 20 wet tons per acre or 2 times the agronomic rate. Samples were collected in triplicate from the 3 different treatment plots and consisted of samples from 1 foot, 3 foot, and 6 foot collected with a soil core auger. And you can kind of see the diagram in the middle kind of shows how we were splitting samples as we were coring down vertically through the soil column. And we collected samples for soil characterization. So it was things like nutrients, metals, physical characteristics, and then also samples specifically for PFAS. And there's the HRSD folks doing the coring. So next slide. Okay, so results that we received, these results are specific for this field. But results from the nationwide study showed that land application of municipal biosolids rarely resulted in unacceptably elevated soil PFAS concentrations, regardless of the land application loading rate. Soil PFAS concentrations were less than or close to soil screening levels calculated using illustrative input parameters. Since SSLs define the maximum soil concentrations that are protective of groundwater, this shows that the potential for significant leaching of PFAS and subsequent groundwater contamination is low at most land application sites across the country. This is also consistent with what we're going to see in the next study looking at Progress Farm. It should be noted that this table, like I said, only shows PFAS compounds that had quantifiable quantities at the Warsaw AREC study site. The soil levels, there's the biosolids are at the top for PFOA and PFOS. And then you can see the breakdown at 1, 3, and 6 feet. And then you have the control, the half agronomic rate, and the 2 times agronomic rate. And ND signifies non-detect. Just going back to the soil screening levels, SSLs for groundwater are risk-based concentrations used by the EPA and states to identify if soil contaminants pose a threat to groundwater quality, focusing on preventing vapor intrusion and direct contact. Using tools like the EPA's RSLs and BISL calculator to set thresholds that trigger further investigation or cleanup, ensuring protection for human health and the environment under different land uses. Next slide. Oh, sorry, hold on, one more, got a little bit more. SSLs are calculated for each PFAS analyte for each individual study site. The SSL calculation takes into consideration soil and groundwater specific characteristics. Essentially, the SSL for a given PFAS analyte at a given site identifies the maximum allowable soil concentration that would maintain groundwater concentrations to less than or equal to the MCL, such as the new EPA drinking water regulation of 4 parts per trillion for PFOS and PFOA. So next slide. To sort of wrap up Dr. Pepper's study, the soil PFAS concentrations at the national level showed that land application of municipal biosolids rarely resulted in unacceptably elevated soil PFAS concentrations, regardless of land application loading rate. These concentrations were less than or close to soil screening levels calculated using illustrative input parameters. And lastly, since the SSLs define the maximum soil concentrations that are protective of groundwater, this shows that the potential for significant leaching of PFAS and subsequent groundwater contamination is low at most land application sites across the country. I would like to add that this phase 1 work has not been published in peer-reviewed journals, but the manuscript is being developed for publication by Dr. Pepper's group. And he did, however, in this report that I showed the link for, utilize a technical advisory committee comprised of other PFAS experts in the field of study. Next slide. All right. So the last study we're going to talk about is an EPA grant from Linda Lee at Purdue University. Dr. Linda Lee is a distinguished professor at Purdue University with a joint appointment in the Department of Agronomy in the College of Agriculture and Environment and Ecological Engineering in the College of Engineering. During her 30+ years at Purdue, she has pioneered research on emerging compounds of concern, including hormones and pharmaceuticals, as well as per- and polyfluoroalkyl substances, or PFAS. Dr. Lee is also a member of the W5170 group, which is the multi-state research group associated with the U.S. Department of Agriculture, focusing on the beneficial reuse of biosolids. She's also a member of the EPA Science Advisory Board, charged with evaluating the framework for the sewage sludge chemical risk assessment. Dr. Lee, this current EPA grant that we're talking about was funded a multifaceted PFAS research project examining the impacts of biosolid operational changes on PFAS diversity and the fate and transport of PFAS analytes associated with land application of biosolids. This has been a long-term project that has examined numerous variables associated with the land application of biosolids and the fate and transport of associated PFAS analytes on rural drinking water systems. HRSD has funded a portion of this work through matching funds to her EPA grant and has provided significant in-kind service for this project. Next slide. The first portion of Dr. Lee's EPA grant examined the changes in PFAS analyte diversity as a result of the thermal hydrolysis process. HRSD implemented a thermal hydrolysis system in an effort to improve the quality of biosolids we produced for land application. Thermal hydrolysis plus anaerobic digestion facilitates the production of higher quality biosolids in terms of percent total solids, vector reduction, and pathogen reduction. PFAS samples for this study were collected prior to the implementation of thermal hydrolysis and post-implementation. PFAS analyte diversity and concentrations were compared and reported. Next slide. Okay, so this is where the chemistry part starts. I know there's a lot of chemistry here, but I'm really just going to kind of hit the high points here. What I want you to focus on is the data from the 2 biosolid processes. There's Class B and Class A. So this study, the results of this study showed that prior to thermal hydrolysis, Class B biosolids contained greater terminal PFAS compound production. Meanwhile, the post-thermal hydrolysis plus anaerobic digestion implementation, Class A biosolids had a reduced percentage of precursor conversion. And so you can kind of see that if you look at the two pie charts on the top. You'll notice that in blue, these are your perfluorocarboxylic acids, which PFOA falls into that group. And then the perfluorosulfonic acids, which are in orange, and this is where PFOS falls. If you notice, you see a significant portion of the total is composed of these terminal PFAS compounds. Meanwhile, if you look at the three graphs on the bottom are post-thermal hydrolysis implementation. Here, you don't see the production of these terminal PFAS compounds. You see everything is sort of stuck. It doesn't go to its terminal state. So you can see that if you look at the perfluorosulfonic acids, or I'm sorry, the fluorotelomere carboxylic acids in gray and the phosphorus containing PFAS in green. You can see that, pre-thermal hydrolysis, post-thermal hydrolysis, and then post-thermal hydrolysis anaerobic digestion, it's mostly composed of these green and gray compounds. This work, it has been published by Dr. Lee's group. It's her PhD student, Catherine Alukow, published this in 2024. So you can go back and look this up. The take-home component, the take-home message from this study, if you go to the next slide, Jamie, is that the PFAS profile in wastewater treatment plants is significantly influenced by input characteristics and treatment process. The thermal hydrolysis plus anaerobic digestion, or Class A biosolid production process, resulted in a 32% reduction in observed quantifiable PFAS compounds. And so what that means is there's reduced precursor conversion. THP did not lead to increases in drinking water regulated PFAS compounds. Next slide. The second portion of Dr. Lee's research examined the fate and transport of PFAS associated with historical Class B land application and then a Class A thermal hydrolysis treated biosolid on the HRSD Progress Farm in Virginia Beach. As I said before, Progress Farm has been the site of biosolid research at HRSD for almost 40 years. It's unique in that there's a long-term historical land application and environmental data set that helps folks like Linda Lee understand the impact of changing biosolids and application strategies on PFAS dynamics. Next slide. All right, so this is a busy slide, but we'll get through it. This portion of Dr. Lee's study provided a valuable data set in regards to PFAS analyte occurrence and concentration in different matrices. And so she looked at rain, biosolids, soil, stormwater runoff, and groundwater. Regarding wet deposition, rainwater samples were collected during 3 different storm events during 2023. Rainwater samples showed significant concentrations of PFOS, with concentrations ranging between 3 to 6 nanograms per liter. These values are approximately equal to current drinking water standards for PFOS. In soils, PFAS compounds seem to be vertically segregated in soil columns, with long-chain PFAS compounds being found in soil surface layers. Meanwhile, short-chained PFAS compounds appeared to move through the soil column more rapidly based on their increased presence in groundwater. It should be noted that in agricultural settings, there are numerous PFAS contamination sources, given their widespread use in the farming industry. PFAS contamination has been found not only in wet and dry atmospheric deposition, but also in herbicides, insecticides, commercial fertilizers, seeds, and treated seed bags. Just like in our residential lives, PFAS can be found everywhere on the farm. Can you go to the next slide, Jamie? I do want to draw your attention to some of the groundwater data. It should be noted that in 4 of the 6 well complexes, 4 of the 6 complexes complied with the EPA standards for PFOA and for drinking water. There were 2 well complexes that did not. But these 2 well complexes are adjacent to an unrelated property with known PFAS groundwater contamination. That is the likely source. Dr. Lee is doing additional work on this to better characterize this potential source. And we'll talk about that here on the next slide real quickly. Next slide. So this is another image of Progress Farm. And here you can see the well complexes. Those are the yellow dots. There's a blue arrow, and this is important because this shows the approximate groundwater transport direction. And then the red circle identifies the 2 well complexes where we saw elevated PFOA concentrations. But just to kind of quickly summarize this, the monitoring wells complexes are located on the perimeter of the property. The average PFOA concentrations in 4 of the wells were less than 2 nanograms per liter. The 2 complexes adjacent to, it's a DOD facility with known PFAS groundwater contamination. The PFOA levels were elevated above the drinking water standards. The averages fell anywhere between 7 and 77 nanograms per liter. And as I said, Dr. Lee is doing further work to better understand this potential PFAS source. And I would like to add that, this is the Ocean Lakes High School community, and all of these folks are on potable water from, there are no drinking water wells out here. So next slide. Okay, to kind of wrap up Dr. Lee's, her current overall findings, her research provides insight on how the diversity of PFAS compounds entering wastewater treatment plants and treatment processes impacts the PFAS content in biosolids. Also, soils play a critical role in the environmental cycling of PFAS through their ability to serve as both reservoirs and channels of transfer into other terrestrial systems. PFAS was mostly retained in soils with high proportions of clay and silt materials, suggesting that restriction of advective flow by fine textured materials enhanced PFAS persistence. Next slide. All right, this is the wrap-up slide. I'd like to say that wastewater utilities in Virginia and around the country are participating and investing in significant research related to PFAS. Source control remains a key focus, and monitoring is supporting a better understanding of the sources to the wastewater facilities. Researchers are actively studying mitigation strategies to further reduce the mobilization of PFAS compounds from land-applied biosolids. HRSD and other Virginia utilities support a structured monitoring program to gain valuable information about PFAS concentrations in biosolids. And that's it. Next slide. So I guess now I kind of went over a little bit, but we have any questions?
It's fine. You had a lot of information to relate to us and to try to condense. I got a couple of questions, but I want to open it up to others in the group. Okay, you talked, let me start with a high level question. What is the status, and you mentioned it as we went along, but what is the status of peer review of the studies that have been completed?
So let's start with the Linda Lee project. The The first part of that work has been published. That I think I stuck the link in there. Yeah, it's the Alocal et al. 2024. I can provide a PDF for that if you want, and you can distribute it
Well,
as you.
one of the things I'm going to ask you is let us have a copy of the slide deck, because we're going to want to post
Okay, yeah.
website, we're going to reuse that on December 3rd, or maybe even a more condensed version. But go ahead. Okay.
And if I can interrupt for just a second, we can put the full citation there just so it's easier to find as well.
Yeah, yeah.
Thank you.
So this part is published and publicly available.
Okay.
The second part of this study is, I think I talked to Linda yesterday, and I believe she's getting ready. It's gone through the first review process, and she got it back for edits, and they're getting ready to submit it later this week. So it is not publicly available yet, but it will be shortly. This part she had to break it up into two sections though. The first part is specifically looking at soils. And then the second part of the study, which we're working on right now, will focus on groundwater concerns related to PFAS.
Okay. While you've got that slide up and you've got that little wet deposition rain cloud up there, did I hear you correctly about the amount of PFAS found?
Yeah,
just
it's right here in the bar chart. Yeah, so that was sort of I mean, it was pretty shocking that, we're looking at a total. This is for, looks like about 8 or 9 different compounds. There were more than this, but these were the ones that were significant. But the total came out to over 6 nanograms per liter, probably closer to like 7, I would say, just eyeballing it.
All right. Pardon me, not being a chemist, and nowhere close. Nanograms per liter, how does that relate to the parts per trillion?
So they're the same. Nanograms per liter equals parts per trillion.
Okay, very good. All right, good. Barbara, you got a question? I've got a couple more, but go ahead, Barbara, please.
I just wanted to say that I've been putting questions in the chat. I don't know if others have or not, but you might find them there.
I can't see the chat right now.
Okay.
Let's see.
All right, we're going to get Jillian to read questions out of the chat then.
Sure. So Barbara asks, according to DEQ, oh, excuse me, that might have just been a comment. Well, maybe it's prefacing something. According to DEQ, most landfills in Virginia send, and forgive me, leachate to a known source of PFAS to POTWs. So far, Virginia source control efforts have not included landfill. I'm sorry, I just want to.
I understand the question.
Okay.
Yes, landfills, they have leachate collection systems, and at least for the ones that I'm familiar with, the leachate it comes to wastewater treatment facilities. Yes, that's correct. At least for the plants I know about. There could be others.
And I can offer a little something here as well. We've partnered with some of the landfills that discharge to us. So what I'm about to say is really specific to HRSD. One of the landfills is putting in evaporation technology, so they're not submitting or sending leachate to us anymore. And the other one we're working with to pilot a foam fractionation treatment technology so that we can so that they, in terms of partnership, we're partnering on the research and development piece and doing a little bit of in-kind monitoring for them. But we're working with them to implement foam fractionation so they can reduce their PFAS load into our facility.
Okay, next, another question in the chat, Jillian.
I don't see another.
Okay, while you're looking, Sarah.
Yeah, I just had a question. So, when you all were talking about Dr. Pepper's research, it seemed like he was concluding that based on the single field that he had tested with different levels of biosolids application, that biosolids application did not necessarily, you know, PFAS were not likely to stay in the ground and contaminate groundwater based on that application and really kind of a single field. And yet we've got Linda's research saying that soil type has a lot of impact on whether or not PFAS are, you know, staying in the soils after biosolid application. Those seem like a bit of contradicting research. Am I hearing that? Or
Well, let me, so I only showed the data from our specific Warsaw study. Dr. Pepper's report includes all 17 study sites from across the country.
23 land applications.
23, sorry, 17
17
states, 23, yeah, sorry. And this SSL calculation, that takes into a lot of different variables. Soil type is one. The application of biosolids is another. Regards to application, you know, we did a 0.5 agronomic rate and then a 2x agronomic rate. So that was sort of the range of biosolid application. And then lastly, one of the components that gets factored in is depth to groundwater. Now for Progress Farm, the depth to groundwater it's probably less than 2 feet. But at study sites, like in Warsaw, for example, you know, we went 6 feet into the soil column, you know, for sampling, and we never hit groundwater. And talking to the AREC director for that facility, I think he said it's around 18 to 20 feet. So the depth to groundwater is another variable. But Dr. Lee's work, you know, she showed that, you know, specific soil type plays a big role. I don't know what the other soil types look like. You know, like if you did a soil core log, was it sandy? Was it silty? Was it, you know, heavy and organic material? You know, it's almost like you have to manage these on a case-by-case basis. Some fields may not be good for biosolid application. Others it may not be a problem at all.
And if I can add, I think what was interesting for me comparing Dr. Pepper's work to the work that was going on in Progress Farm, and he was looking specifically at the regulated contaminants too, with PFOA and PFOS from a drinking water standard. That work did mirror what we saw at Progress Farm with Linda's work. We really weren't seeing that transport of PFOA and PFOS into the groundwater as a result of that land application. We did have PFOA showing up in 2 wells, but they were right next to contaminated groundwater from adjacent property. We know the direction of flow of groundwater at that site. And so what was, again, interesting to us and aligned with Dr. Pepper's study is that transport and mobility of PFOA through the soils wasn't creating a drinking water concern from a regulated standpoint.
Hey, Jamie, can you jump to slide 24? I mean, it's pertinent to what you were just saying.
Here we go.
One more.
Oh, one more.
Okay, so no, 24. What, back one?
Oh, sorry. We were out of sync in terms of timing.
Oh, okay.
There we go.
The red circle is the 2 wells that showed the highest levels.
Yeah.
Okay. And so the DOD facility is off the bottom of this picture.
It's to the east of this property.
Okay, well, I don't know where north is in this picture.
Yeah, sorry. It's
to the
actually
right.
oriented. Yeah, it's oriented. So it's this way. This is the property.
Okay, so it's the right.
Yeah,
right.
this whole thing is the property over here.
Okay, so it is directly upstream of the groundwater flow.
Yeah.
Okay.
Downstream. So
Our wells are downstream.
Yes, the property is upstream.
We're talking about a different piece of property. Okay.
Yes, the property is upstream. Yeah.
Okay, well, that was one of my questions, so thank you.
We have
Did something else come in on the chat?
Yep. Barbara has another question. Did Dr. Pepper's research, excuse me, include mass balance calculation and or geochemical analysis. Given the precipitation component, did Dr. Pepper's work include pre-application testing? She notes it's important to include comparison to the MCLG levels as well as MCLs in future talks to more accurately indicate human health risks.
We did do pre-application. I didn't add it to the slide, but you can get an estimate of what the pre-application looks like or looked like if you look at the control sites, the control plots. So it's, for the PFOA and then PFOS, if you looked at the 1 foot depth, that would kind of give you an idea of what the pre-application concentrations look like for those two compounds.
Hey, Chris, that control site, now it was a control site for this study. Had that been part of an area that was used for various
testing? It had never, so if you go to the aerial, the control site is in, sort of the red dots. This plot had never received biosolids.
Never.
Okay.
All right, good. Thank you. Now, another one of my questions. We're all kind of a similar path here. Let's see. You had said, we're on, and I did not write down the slide number. You used the term rarely in your description, and can you add a little more specificity to what?
So this is a quote from Dr. Pepper's report. Rarely resulted in unacceptably elevated soil PFAS concentrations. There were a few in the report. There are, I'd say, less than a handful of cases where the PFAS concentrations were above acceptable levels. I can't remember the exact number for that, but he's got a lot of data in his report that's available online.
Okay, so when you say a handful out versus what, 100, 200?
You'd have to. I can't.
Oh, come on.
Quantify that off the top of my head.
Okay, fair enough.
This came from his conclusions.
Okay.
So
now and this has not been peer reviewed, right?
It has not been peer reviewed.
Where do they stand in that process?
They are, the technical advisory committee is currently reviewing it.
Okay.
Once they are finished reviewing it, then he will submit it to the peer reviewed journal process.
Okay, thank you. Jillian, do we have other questions in the chat? Because we've almost run through all mine.
I've seen a bunch come through.
Yep. So Barbara asks, our region of Virginia is underlain by karst. Is there any research on karstic soils? Again, apologies if the pronunciation is wrong.
I don't know if they've done specific studies to compare karst ecosystems to non-karst systems. I'm not a geologist. I know that Dr. Lee, sorry?
Barbara is.
Oh, okay. So Dr. Lee has, she's doing the study at Progress Farm. She's got a couple of other sites near Purdue in Indiana, and then she's got some more scattered in some other spots around the country. In Dr. Pepper's study, I don't remember him explicitly comparing like karst to other types of soil systems. So that's a good question. I don't know the answer to that.
Yeah, I don't have a precise answer or a complete answer, but I know that Virginia Tech was working on doing similar research at the Agricultural Extension Center out near Blacksburg. And I think that was in collaboration with another utility to obtain the solids for land application there. But I think that's still in the early stages, but we can follow up and get you more information on the status and the work that's going on there. So I think that could, that the outcome of that work could address some of that issue, because I'm assuming that's karst topography out in that area.
And then I see Barbara has one more. Could you say a little more about the biosolids that were used in the studies? How typical are they? So that's all. Yeah, go
so
ahead.
these were DC Water's Bloom biosolids, and the biosolids, if you go back to the data slide, the data table.
Sorry, I was going the wrong direction.
I'm sorry. Yeah.
Right here.
I don't have off the top of my head any of the sort of physical data from this, just the PFOA, PFOS concentrations. But these were wet biosolids. That's a Chris Piot question. They were more than likely, I'd have to assume that they were representative of their normal production quality control standards.
And I can comment on these values compared to what I've seen from a lot of other residential municipal biosolids. And these are fairly consistent numbers. Typically we see 25 parts per billion or less, usually, in these municipal biosolids. And so that's certainly what we're seeing here. I think what's interesting, just to take it back to Progress Farm, is that, with the historical land application that occurred there over the decades, that historical land application would have occurred during periods of time in which there were no controls on PFOA and PFOS. And so one might presume that those concentrations were certainly a lot more elevated, say 15, 20 years ago during those land application periods. And so to me, the groundwater data is extremely encouraging based on that. To some of the comments before, I think in Dr. Pepper's work and Dr. Lee's work is all consistent. These are site-specific management issues, and I think the site-specific conditions in the soils matter. The organic content of the soils is pretty important, and that's what we've seen from the draft risk assessment work from EPA too. So I would envision that as EPA eventually moves to creating regulatory standards around these, that the mitigation practices that we would employ for land application would include things like site-specific characteristics of soils, organic content, things like that, because that does matter in terms of mobility of the PFOA and PFOS. The other thing that I think is interesting that you might also find intriguing is that HRSD is working to see if there are things that we can incorporate into our biosolids to further reduce the mobilization of PFOA and PFOS. And so we've worked in the past on some biochar blends, and we're working now with another biochar blend to see if that decreases mobilization. But again, early in the study, but that work is work that's characteristic of work going on all across the country, just trying to find ways that we can further mitigate risk with the land application of biosolids.
I've seen a couple of other questions come through.
Yeah.
We have maybe a more of a note from Evan Isaacson. Last year, Dr. Pepper joined a handful of Penn State scientists to give a webinar on PFAS in biosolids. His presentation, which Isaac said that he can share, or Evan, excuse me, showed substantial increases in the concentration of many analytes. That was a nationwide project, and according to the presentation, also included sites in Virginia. Not sure how to reconcile what I've heard today versus what he presented last year with the colleagues at Penn State. Evan can share that presentation by email when he gets back to his desk later, joining by phone.
Thank you. And then let's see, Stuart Overby had a couple of questions. Can you see those, Jamie?
I cannot stop sharing to see them.
How long?
Oh, sorry.
How long the time of application and how long between the time of application and the time of sampling, and why?
That was approximately 12 months.
Okay. And why was there no sampling of soil above 1 foot depth? If you go back to that I can't answer. So the 1 foot sample was really, so the first 6 inches we discarded, or actually we didn't discard it. We actually scraped the surface off, and then we collected, you see the red dot
That went to the HRSD analytical lab, where we looked at things like nutrients and just sort of general physical chemical characterization. But then the first PFAS sample was a composite of the 6 to looks like 6 to 18 inches. So we composited 12 inches of soil material for that 1 foot PFAS sample.
Okay.
And then you can kind of see that from 18 to 30 was another composite that went to the analytical lab. And then from 30 to 42, we collected another PFAS sample. And then we discarded another 12 inches of sample. And, you know, you can kind of see the dichotomy of how we split up the soil cores.
Okay, let's see. Would you say that pre-testing of soils would be prudent before adding more biosolids?
I would always collect a pre-application sample, just because you don't know what the history of a field is. You don't know, PFAS is everywhere. It's not just in our homes, but it's also used in, you know, PFAS compounds are used in insecticides, herbicides, inorganic fertilizers. It's used in the containers or the bags that contain seeds. It's everywhere. And so if you're going to try and characterize and understand how PFAS is impacting a farm site, you really need to have information pre-application. I just think that's imperative.
And I think that that's from a research perspective, right? So if you're trying to do a research study and understand the impact of biosolids and potential soil concentrations, then having that characterization data before you put it on the property, that's really important in terms of research. And, you know, this control plot here, we know that at Warsaw, they did not have any biosolids land applications. That control plot does a pretty good job of letting us know what that property looked like ahead of time. In terms of land application and characterizing biosolids concentrations of PFAS, it's really important to do that at the site of the generator, because we can't control, and we certainly won't understand specific biosolids related PFAS if we do that on the field. It's understanding concentrations of PFAS and biosolids really requires that we do that PFAS monitoring at the site of the generator.
Barbara has another question. Did any of the studies include surface water and sediment testing, vegetation uptake, or fish and wildlife uptake? I think there was discussion.
I can answer that question.
Uptake in one of the studies.
So we just collected soybean samples for the Ian Pepper study back in late September, early October. Those samples have been shipped off for analysis. Dr. Lee has sampled, at Progress Farm, we've had a couple of different crops, corn and soybeans, and we've sent her plant samples for both of those seasons of growth. That's something else that's also important for PFAS research, is to understand how different crops, you know, how do they grow? You know, what does their root material look like? And then some corn versus soybeans. I'm not a botanist either, so they the processing of these nutrients and chemicals, it can be different between the different plant species. So that we have sampled for that. We do not have data yet for that. I think you also asked about surface water. For this particular study site, there were no surface water samples available. If any of you have been out to the Warsaw AREC, it's just a big field out in the middle of nowhere. There at the Progress Farm, we have sampled in the lake. If you go to the map of Progress Farm, okay. So if you look here, you can see that sort of in the lower right-hand corner, that's Lake Tecumseh. We have done sampling
which
there.
Is between the DoD site and your site.
Yes. So this is a lake that has been here for forever. But so we've done some sampling on this lake, and it wasn't until we saw the PFAS issues in these two wells that we really started to ask, like, well, where is this material coming from? Because, you know, all the other wells have negligible PFOA concentrations. And so then we went to the DoD facility, the folks that run that, and there was a wastewater treatment plant that used to discharge into this lake prior to 1972. They did a lot of firefighter training at this facility. And, you know, I don't know the specifics of the wastewater treatment plant, but I imagine that there was, you know, they were using AFFF. So there was a PFAS source there. There's also an unlined landfill that is sort of between the lake and the Atlantic Ocean, that also they disposed of firefighter training equipment and residues. So we don't know that we can really trust, you know, the surface water is fine. There's really no measurable PFAS in the surface water. But I suspect once we go into the benthic cores in the lake, that we'll start to see evidence of this historical use. Does that answer your question, Barbara?
And then she's got a follow-up.
Okay. Oh, what was,
how do these studies compare to the extensive biosolids, farm, soil, crop, and livestock, and other PFAS testing done by the state of Maine?
Yeah,
okay.
I can speak to that a little bit. I think what's important about the Maine work, they did show PFAS contamination, but the biosolid source wasn't a domestic biosolid source. That those fields had also been applied by industrial sludge from paper mills, pulp and paper, and we know that those are PFAS contaminated solids. So there's been a conflation between solid sources and descriptions of the land application that was occurring in Maine. There are industrially impacted solids, and those can contribute to PFAS issues on a property, but there's not a lot of industrially contaminated biosolids. Biosolids largely reflect residential use from municipal wastewater agencies. Sludge from industrial processes like pulp and paper mill are typically higher in PFAS concentrations.
And we heard similar in our presentation by Southern Environmental Law Center, looking at industrial sources and some of their experience in North Carolina, and the highly effective strategy of going after the source, and before it gets into the wastewater treatment. Okay, one last question from Barbara, and then I'm gonna wrap us up. Are the wells all completed in the same hydrologic unit? Good question.
The answer is yes. They are, there's, in most of the well complexes, there's two wells. There's one that's drilled to 35 feet with a screening at, I think it's like the bottom 5 to 10 feet. And then a second well to 50 feet. These wells were put in to look at potential nutrient concerns, you know, 40 years ago when this farm was started, or it wasn't started, when the research was started there. All of these wells are in an unconfined surface aquifer. So it's, I hope that answers your question.
Yep. Okay. Jamie and Chris, thank you very much.
Yeah, if I can just kind of close out, if you don't mind, just a moment. I just want to circle back to, you know, there are really just 3 management options. We produce this material every day. The only 3 options really are land application, recycling, incineration, and landfill. And we really need a diversified portfolio of management options. And I think we're possible, I think really prioritizing the productive use of that end product. And so returning again those nutrients and that organic matter back to the soils. There are farmers that really appreciate getting this material, reduces their costs on the property, really helps with their crop production and yields. And so I think really trying to maintain that connection to the farmers is important. So all three of these options need to remain as viable options within the state. And I think having these this portfolio of strategies is critical.
Excellent wrap up. Obviously, for the commission meeting on December 3rd, we can't go into this degree of detail, and that's what the technical committee is for. I appreciate everybody's questions. Barbara, thank you. And thanks, Stuart. She had to leave for another meeting. So did Pat. I know where they are. They are watching the Water Commission, who scheduled a meeting for 10 o'clock. We weren't concerned with conflicting with our meeting, but they're hearing about the Chesapeake Bay Watershed Agreement refresh. But then they're going to talk about rainwater harvesting, and then they're going to talk about proposed legislation on these topics. So when we complete this thing, I'm walking across the street to the Capitol to hear that, and we can talk more about it in our future discussions. You have on your agenda, folks, the proposed meeting schedule for 26. As we always do, we suspend after this November meeting because of the holidays, and then General Assembly session. We'll pick things back up in March. The commission meeting next week, we'll be having another presentation on the other of our favorite recent topics, data centers. But then the Southern Environmental Law Center, Carol Courtenay, and then Jamie and Chris here will be reprising briefly their the presentations that we've spent in both of our last two meetings well over an hour on, and it's much appreciated. We'll be meeting at our normal December site on the 3rd, which is Meadow Event Park there in the State Fair area in Southern Caroline County. All are welcome to attend and or welcome to request the link from me. Just send me an email and I'll get that to you. I'll probably send that early next week. I really appreciate the time that people have dedicated to provide us input on these presentations. It's been very helpful. You know, I'm an urban planner. Jillian is a public policy person, so the chemistry is very challenging. But you know, we're working at it. Any comments from committee members before we wrap this up? Okay, thanks again, everybody. I hope everybody has a great Thanksgiving. Let's see, Deirdre and John. It doesn't look like John is still here. Anyway, Deirdre, you've been doing this with me for a long, long time. So enjoy your holidays. We'll pick it up in 26.
Thank you all for being generous with your time. We really appreciate it.
And no, great presentation, a lot of good information, and looking forward to, as this research develops, hearing more, especially as they reach peer review. It'll be helpful in the public policy development that is coming.
Agreed. Thank you.
All right. And this will be, the recording of this will be posted here in a few days. We'll get your slides up as well once you send them to us, if you want to add those links, and so people can go to those studies if they wish. All right.
Yeah. Is by the end of this week okay for you?
Oh, absolutely.
Okay, good.
Yeah, yeah, it's holiday season. Nothing happens. All right. Thanks again, everybody. Appreciate the new participants this time.
Thank you.
Have a good one.
Thank you.
Bye-bye.
Hey, Barbara, since you're still on, can you come off mute? No, maybe not. All right. Michelle, good luck with your meeting tomorrow.